Primary antibodies are created to detect your target of interest, and they can be polyclonal, monoclonal, or recombinant. Let's start talking about the polyclonal antibodies. We already discussed that antibodies are generated by B cells in the blood serum. Each antibody is generated by a specific B cell in the blood. Each one of those antibodies is called a clone. That's why these antibodies are called polyclonal, because they come from several different B cells. Different hosts can be used in the production of antibodies. I was actually mesmerized when I learned how many different hosts can be used to produce antibodies. I was in high school when I was trying to determine where I wanted my career to go, when a friend of the family who was a scientific researcher decided to take me to his lab, and he worked on vaccine production. And that's when I learned that different hosts can be used depending on the purpose for which that antibody is going to be used. Depending on their antibody, they would use mice, rabbit, sheep, goat, donkey, and so on. So that was actually one of the cool experiences I had as a kid that definitely helped me decide to be a scientist. They didn't let me pet those animals, but that's a whole different story. Once you have determined which antigen you want to inject in that animal, you're going to treat the animal with that antigen, and then you're going to wait a period of 2 to 3 weeks so that the animal can produce an antibody, much like what we do when we're fighting a disease. So we wait that period and we draw some blood. At that point, we look at how much antibody is present in that animal's blood. If it is enough, we're ready to proceed, but more than likely we're going to have to give a booster injection, which is injecting the same animal with the same antigen. Then we wait another 2-3 weeks and repeat the process until we have enough antibody on that blood serum. At that point, we collect that serum and we move forward with antibody purification. Now, because IgG is the most common isotype of antibody present in the blood serum, that is the one that you're going to find commercially available for polyclonal antibodies. Some of the advantages of a polyclonal antibody are they're easy, fast, and cheap to produce. They're also fairly easy to produce in large scales. Because you're going to have several antibodies targeting one antigen, you can use them for several different applications. On the counter side, some of disadvantages of the polyclonal antibody are they can have high cross-reactivity, there can be lot-to-lot variability since each time you are injecting an animal with that particular antigen, and because we have more than one antibody targeting that particular antigen, that primary antibody may also be less specific.